give me the 10-minute warning is cool. Yeah. All right. Okay, so hi, I'm Marian. I'm a first year PhD student at Geoscience. Before I start, I'd like to acknowledge and pay respect to the Widjabul people of the Bundjalung Nation, from whose land I'm presenting today. This presentation lays the foundation for what you can expect from my PhD, as well as a background and overview of how we can use isotopes in the archaeological record. So to start, I'm going to explain the whys and whats of isotope analysis in teeth, the kind of methodology I'll be using, the sort of data we can get, and how that data can be interpreted to create a fascinating narrative. And then I'll introduce the star of the show, Gigantopithecus, and explain why I'm undertaking isotope analysis on this enigmatic species. At the heart of this story, though, it's about the largest ape to ever live, its teeth, the information they contain, and the mystery surrounding its extinction. So isotopes are present everywhere in the world in which we live and breathe, but the balance, or the ratios in which different isotopes of the same elements occur, varies between different substances, i.e. different types of food, or between ecosystems, i.e. land or sea, or even different climate zones. As we grow and continually as our tissues develop and renew, the isotopes that are in the food we eat and the water we drink are being incorporated into all our body tissue, including our skeleton. By measuring the ratios of different isotopes in bones or teeth, we can find out how they occurred in nature and trace them back to the source from which they came. So then archaeologists can find out many things about the individual, such as what their diet was like and the environment in which they grew up. There are many stable isotopes that are used by archaeologists, but some that are most widely analysed are carbon. We use the delta 13 carbon, nitrogen, delta 15 nitrogen, oxygen, delta 18 oxygen, or the strontium 87-86 ratio. Carbon and nitrogen stable isotope ratios are most often used to explore a wide range of questions surrounding diet, while the main application of oxygen and strontium isotope ratios is to reconstruct ancient migration. Stable carbon and oxygen isotope analysis of human and animal tooth enamel carbonate has been applied in paleodietary, palaeoecological, and paleoenvironmental research from the recent historical periods back to over 10 million years ago. Strontium ratios have been used in a range of paleomobility research, as well as the more recent historic, and can accurately trace migration through seasonality, provenance, food, and determine whether an animal is local or not local to a site. In my PhD specifically, I am focusing on carbon, oxygen, and strontium stable isotope ratios, as well as trace element analysis. So one of the reasons we use teeth is because tooth enamel is primarily made up of biapatite crystals, which are larger than those in bone, which means it's more resistant to post-mortem diagenetic ionic substitutions and contamination. A more practical reason, though, is that often they're all that's left of the skeleton. In the case of Gigantopithecus, porcupines have gnawed away most of the remains to get to the marrow, but have to leave the enamel tooth caps because they're so hard. So sometimes that's all that's left. In this photo, you can see me preparing the teeth for laser ablation. I slice the tooth at its earliest cusp because I want to make sure I'm getting the most information about the early life of the fauna. I follow this by polishing it to a flat surface so the laser can be consistent in its ablation and focus. The tooth will then undergo several types of analysis. The first technique, as I mentioned, is laser ablation. This is coupled to a multi-collector inductively coupled plasma mass spectrometer, or a quadrupole. The laser will ablate the enamel and it will then be sent first to the quadrupole for trace element analysis and then to the multi-collector for isotope ratio analysis. This will give you two different types of data. The first, an isoscape pictured up on the top right. This is a trace element map, which I model in RStudio. It provides a visual representation of the movement and location of elements within the tooth. The second, at the bottom, is incremental sampling. This is one of the really crucial stages for my PhD. Using strontium and incremental sampling technique, I can determine the mobility of an animal over time and in reaction to climate. I can see whether they were originally local to that site and see their seasonal migratory routes. The next technique is using solution chemistry on the same samples to compare the accuracy of my laser values. Traditionally, the sample solution is coupled with MC-ICPMS, which is more accurate than laser. So I just want to make sure I'm accounting for and correcting for any problems my laser has. But laser is so much faster than solution, I really need to do it to get the most out of my results. The third technique is the analysis of the delta carbon and delta oxygen in tooth enamel. Given that tooth enamel forms incrementally over its period of formation, it can be sampled in bulk along the entire growth axis or sampled sequentially in order to investigate the changes in diet and environment that have occurred during the formation period. Chronologically ordered dietary change within a given tooth is observable for humans and other animals. It provides information regarding the interannual, seasonal, and dietary variation. These methodologies have been widely applied and described in archaeology, ecology, and paleontology. And it's especially interesting in the case of Gigantopithecus because we don't know how they adapted or didn't to their changing climate. And on that note, I'd like to introduce the star, Gigantopithecus blacki. No, it's not Bigfoot or Sasquatch. Giganto has been used in cryptozoology circles as the identity of the Tibetan Yeti or American Bigfoot, human-like monsters in folklore. To this day, Giganto is still used as an example within the monster spotting societies as evidence for their existence. However, the fact is, Gigantopithecus is an extinct genus of ape that lived from the early to middle Pleistocene in southern China and northern Vietnam. Uniquely, it's the only ape to become extinct in the last 2.6 million years. The first remains of Gigantopithecus, 2/3 molar teeth, were identified in a drugstore in China by the anthropologist Ralph von Koenigswald in 1935, who subsequently realized they were primate teeth. Gigantopithecus has traditionally been restored as a massive gorilla-like ape, potentially 200 to 300 kilograms when alive, but the paucity of remains has made it difficult to make total size estimates. What we do know about their diet is that Gigantopithecus was a herbivore. Carbon-13 isotope analysis suggests consumption of C3 plants, such as fruits, leaves, and other forest plants. Most of the analysis undertaken on giganto teeth, however, has been looking at dental calculus and micro or mesowear. To date, only 17 teeth have undergone isotope analysis, other than for dating. While only being a distant relative that branched from the human lineage around 12 million years ago, Gigantopithecus blacki was part of the lineage that evolved into orangutans, with gorillas and chimpanzees being our closest primate relatives. More importantly though, they occupied southern Asia at the same time that Homo erectus occupied Java and the small-bodied hominid occupied Flores in Southeast Asia. And this may have been subjected to the similar evolutionary and environmental pressures. However, while Gigantopithecus blacki experienced severe range reduction and extinction in southern China, Homo erectus and archaic humans expanded their range and survived. Giganto probably evolved within the subtropical environments of southern China, becoming highly specialized and endemic to this region, while Homo erectus evolved outside of this region, preferring the open woodland and grasslands, but was able to adapt, occupy, and survive. So the question is, why did the ape fail and humans persevere? And what does this mean for primate and human survival? Well, my PhD is a larger project seeking to answer just this. I specifically am looking at 11 sites, which are right here, my PhD sites. And 10 of these contain Gigantopithecus teeth. These sites span the known existence of Giganto, so I'm looking at a range of between up to 2.3 million years to about 300,000 years ago. I'm undertaking a comprehensive suite of isotope and trace element analysis on teeth. So far, there's been no comparison of past diet and past environmental records from the same cave sites. Previous Gigantopithecus studies have incorporated only one of these techniques, but neither have been both used to provide a context for primate activity alongside past environmental conditions at the same cave. The past diet information from stable isotopes and trace element analysis of the teeth will reveal what vegetation Gigantopithecus was eating at the time of its extinction, as well as its mobility and migration patterns. This information can be directly compared to availability of those types of vegetations, according to the environmental records, which will reveal whether a habitat deterioration or a collapse could have been one of the potential causes for their extinction. Their mobility will also do the same. Being compared against a climate change record in the environment, we can see,
Marian.
Okay, no worries. We can see where they moved, how they moved, and how all of this ties together, and see their reaction to climate change and whether this is what caused their extinction. Okay, that's it. And this is acknowledging my supervisors who are here. I've got Dr. Renaud and Professor Anya here at Southern Cross. Ian is in Adelaide, Kira Westaway is at Macquarie. She's actually the CI of this entire ARC project. And then this is a much bigger project called the Giganto project. So yeah, please have questions.
Excellent. Thank you so much, Marian. That was really, really interesting. Anyone have a question for Marian? Belinda?
Hi. Sorry, I'm actually in Adelaide, so Marian invited me. Thanks, Marian. Marian, I'm just really blown away of how much you've progressed in this year. Like, it's been such a horrific year, and sitting in isolation and learning all this on your own. I'm just so proud.
Oh.
I guess, like, I've got two things. There's two things I've never really understood with isotopes is, like, how exactly you take something from the tooth and then be able to talk about mobility and things like that. And then the other thing is, like, did the advent of Homo erectus or the what was the other one? The
Flores.
Yeah, or Flores or anything like that have any influence over Gigantor disappearing?
Okay, so I'll answer the isotope question first. So with the mobility, the best way to think about it with strontium, for instance, is you are what you eat. So strontium is in the geology wherever you are. And that means it's in the water, the plants, the trees, and everything you might consume. And so it's bioavailable. So it starts the lowest process down in the soil, and it gets brought up into plants, which animals might eat or we might eat. We might then eat those animals. And so it stays in our teeth because strontium will substitute for calcium in the tooth. So effectively, if I was going to give you an analogy, I would use a beer analogy in Australia, right? So in Western Australia, we're a big fan of like emu bitters. And then in Adelaide, you might think of Coopers. In Victoria, you might think of VB, right? So let's say you're a child who drinks beer, which is bad, but let's just say you are. You start in Western Australia and you're drinking that emu bitters. There's a record of that in your tooth. And we know that emu is Western Australia, so we can see that. Then you move to Adelaide because you move a lot as a kid and you start drinking a whole lot of Coopers. And then we see the Coopers beer in that tooth as well, because we know that that Coopers value is directly associated with the value in Adelaide. And then you move again to Victoria and you start drinking VB. And so when I take the tooth and I start sampling along it, I'm going to have a direct record of the exact values for emu bitters in Western Australia, where I know it comes from Coopers in Adelaide, where I know that comes from, and Victoria, because I know that VB comes from there. And the same is with strontium. It has specific ratios for every type of geology. And so we have a map of most geologies, and I can then compare. I can say, okay, well, this tooth has a range of these strontium values. It's probably been to these local geologies in its area, and then I can trace where it's been moving. So that's the strontium mobility. I hope that kind of helped. In terms of the impact the Homo erectus or Homo floresiensis had on Gigantopithecus extinction, we don't actually know really too much. Most of what we have is a bit of an educated guess. So we don't see any association with Homo erectus and Gigantopithecus, but that's not necessarily mean there isn't any, because a lot of the gigantoteeth we found have been moved by porcupines into these caves. So we don't actually know necessarily where they were living and whether they were interacting at all with Homo erectus. What we do know is that there are no skeletal remains that have been found, although there's not many, that have any kind of butchery marks or hunting marks. So we don't think there was necessarily that kind of interaction. The other, thing you might wonder is whether Homo erectus was putting resource pressure on Gigantopithecus. The answer is probably not, only because Gigantopithecus had a very sort of specific type of vegetation range, kind of similar to panda, in that panda really only eats bamboo. Giganto really only seems to have eaten those kind of fruits and sort of soft vegetation. And while Homo erectus might have been as well, it's unlikely that there would have been that much pressure to drive Giganto away from that area. More likely is the change from that sort of tropical environment to a more savanna environment, which Homo erectus could adapt to, but perhaps Giganto couldn't.
I have a question.
Yep.
So when we think about the isotopes and the Paleolithic diet of humans, what do you think the implications are on that, considering how our diets now are very similar and highly processed, and like we're not eating anything that's very close to our old Paleolithic way? And what sort of impacts could that have? What
it means for sort of mobility research, for instance, is that actually modern samples are really not able to have that kind of research done, because nowadays, in the last probably 100 years at least, growing up, we're kind of eating things from all over the world. And so that strontium signature in our tooth is now no longer reflective of our local environment. Where sort of the limit now for strontium mobility research is kind of in what we'd consider like that historical period, right? Where people really didn't range far outside their local areas. So we can still look at it for, say, at least in the 19th century, we can still get those values. People moving to Australia from England, we can see that shift. But now it's not really too useful for modern humans. What it is useful for, though, is like provenancing food or wine, actually. And you can still see those differences in the types of food. You just can't really see it in modern humans anymore.
And what sort of influence is that going to have, or impact is that going to have for future researchers in another 100 or so years?
Well, hopefully nowadays we have so many records of everything. Everything's like written down or recorded or in video. There's kind of no secrets about this time period. So hopefully we wouldn't even need this type of isotope analysis on modern humans, because it's kind of already there, that understanding.
That makes sense. Thank you, Marian. Does anyone else have a question?
Yeah, I could maybe ask one.
Go for it.
Yeah, interesting presentation. It's really great. I'm just sort of, I don't really know how to contextualize it, but what are like the main drivers of this research? Is the main driver of this sort of work like an interest of our history, because we're obviously interested in our history, or is there something we take away from this that helps us move forward or something? Or I was just wondering, what are the main like drivers of this kind of research?
It's a bit of both, but what I guess you would say in kind of what's the use of it in a more modern context outside of like interest in knowing our past is really seeing those adaptations to climate change, and seeing why certain species adapt and others don't. And this has more of an impact than just looking at humans. This is kind of like an ecological study too. We're seeing those changes in paleo environments and why and how different species seem to survive it and others don't. So it's kind of that. Yeah.
Perfect. Thank you.
No worries.
Does anyone else have a question?
Hello.
Hey, Marian. I was just wondering, how big is your map of geology?
So it depends. Different researchers or PhDs or postdocs have sort of mapped different areas. So Southeast Asia is still undergoing a lot of mapping. Actually, one of the PhD students on this project is doing a lot of that mapping currently. We have full maps for a lot of Europe. Some of Australia, most of Queensland has been done, most of Western Australia has been done. It's not a particularly difficult thing to do, it's just time consuming because you have to take soil or plant samples from all the different areas and then run that isotope analysis on it. But there's heaps of people working on it, and it's like got like this big open access data sharing platform that we can all access those maps. Yeah. So.
Great. Anyone else have a question?
I'm just checking the app for you, Marian.
Oh, thanks.
There's none in the Zoom chat.
Yeah, you've written in there, feel free to ask me anything isotopes or archaeology related. No one's popped anything in there as yet, but people will probably come to that when they get a bit of time to Marian.
Yeah, no worries.
And you can also
I loved your presentation.
Yeah, you can also put questions down if you watch the video. Like, people will watch the video and ask questions, so just check your Hoover app over the coming week or so.
Sure.
Excellent. Well, thank you. I want to say thank you to all the presenters today. All the presentations were really, really interesting, and it's excellent to expand our views on things that we are not experts in or have any knowledge of. Yeah, so thank you, everybody. Was there anything else that we needed to do, Leslie, before we went today?
Well, obviously, we'd love to encourage everyone to come to our next session, which is